In this tutorial we will discuss the gene selection module. As you can see, a project has been loaded and run through the HTO demultiplexing module and the quality control module. The gene selection module aims to reduce the number of genes used for future analysis. You can see that the project currently contains over 18,000 genes. This number of genes is too large to carry out the successive analyses. The aim is therefore to find the most informative genes for the analyses. Several methods are available to find these genes. If we use the VST method, we can ask for a number of genes to be selected. Let's ask for 1000 genes and click on the button to start the analysis process. Once the analysis is done, we get several results. Firstly, a table showing the genes that have been selected. This table contains the name of the gene, the average expression of the gene across all cells, the percentage of cells expressing that gene and the standardized variance of the expression of that gene. This table only shows the first 200 genes out of the 1000 that were requested. The complete result can be exported to a file using the download button. The second result is a scatter plot that shows the average expression of the genes and their standardized variance. Each point is a gene, and the most variable genes are shown with their names, but all the genes in red are selected, while the genes in black are not retained. We can see that there are 1000 genes selected and 17,244 genes not selected. We can use another method, for example the minute var plot method. This method does not ask for a number of genes, it calculates it by itself. We can start the process by clicking on the button. Again, the result is a table listing the first 200 genes selected and a scatter plot here showing the average expression and dispersion. As can be seen, only 447 genes were selected. Following the creation of these gene lists, you can access the Gene List module. This module allows you to identify and edit the different lists you have created. These gene lists are all listed here. By default, a gene list created from the Gene Selection module has the name of the project, followed by the name of the method and the number of genes selected. Here FL tutorial after DMux after QC is the project name. VST is the method name and 1000 is the number of selected genes. By clicking on this list, one can see the genes that are present in the list and modify the list by selecting and deselecting some of the selected genes. This allows the list to be refined according to the needs of the analyses. Once the gene selection has been applied, we can move on to the next module, which consists of PCA and regression. You will find a video tutorial on this subject.